Great, thank you. I sincerely appreciate the opportunity to give a second talk. I want to present on another potential method to eradicate fish, invasive fish. Obviously, this is a very important topic in Hawaiian Anchialine pools, so I'm happy to have the chance to present some of the work that we have done. I just want to acknowledge the collaborators, Maya Munstermann, Malia of Hōnaunau, Carter Atkinson, Cupp, and Banko. So carbon dioxide has a long history of use in fisheries management. It was originally used as an anesthetizing agent to handle game fish, for handling and transportation, things like that. But more recently, it's been applied for invasive fish control. Just as a reminder in the chemistry, carbon dioxide, when it's mixed in water, forms carbonic acid, and then that disassociates into free hydrogen and carbonate ions, and those free hydrogen ions, the more of them in the water, the lower the pH is. And when you have CO2 and low pH, it decreases the oxygen uptake in the fish. And that can cause respiratory acidosis and oxygen debt. And at low concentration or short duration, then it anesthetizes the fish or kind of stuns them. But at higher concentration or longer duration, it can result in mortality. And just some examples from Aaron Cupp's research on the mainland he's been working on is for lethal treatment for control of invasive carps. And you can see on the right, those are two photographs where they applied CO2 into these large artificial ponds that was capped by ice, which is like a perfect situation so that carbon dioxide is kind of trapped under there over the winter. But they've also used behavioral deterrents, and again for invasive carps and round gobies and sea lampreys and things like that. And can you manipulate their behavior, or can you set up these barriers that will prevent movement up like the Mississippi waterway in the continental U.S.? So our aim was to see if carbon dioxide can be used to eradicate invasive fishes in Anchialine pools. And of course, we already know the main players are the tilapia, guppies, and western mosquito fish, but there are a few others. As Lisa Merrick and her folks have shown that up to 25% or so of 400 plus pools they looked at had invasive fish. Clearly, it's a pervasive problem in Hawaii, impacts native invertebrates. We all know that. And I think what this does potentially is it provides an alternative to, say, rope known or a hu'u or some mechanical method of collecting. And I think the more weapons basically we can have in the arsenal for managers, the better, because there are different priorities and different constraints that they have. So again, the more options we have, for sure, the better. I would say it's not free, but it's relatively cheap to use, particularly once you have the equipment, and it is very simple to use. So our goal was to test the efficacy of the CO2. First, we conducted aquarium trials, and then we conducted field applications as proof of concept. So our aquarium trials, yes, we tested on those 3 fish, but we also tested on potential non-targets, the feeble shrimp, the opae'ula, and a theriid snail. And based on kind of previous work, we identified 5.3 as a target for tilapia, but also we tested 5.0 and then a pH of 5.0 for the guppies and western mosquito fish. And we treated for 2 hours over 2 treatment periods, but the 2-hour treatment periods were interrupted with a 2-hour, what we call a recovery period, where they were put back into unaffected water. And that was designed to kind of mimic a tidal flow. It's shorter than a typical tidal flow, but we figured if they survive with a 2-hour recovery period, then they're probably going to survive with a longer, like 6-hour recovery period. We observed body response, breathing response, and position in the water column, and we thought that these behavioral observations were important because as fish are being removed from the pools, how the CO2 affects their behavior could facilitate our ability to collect them, or the management strategy for trying to collect them. So the behavior was important. And then we looked at survival. Apologize, there's a lot of information on here, but kind of think of this in really broad strokes and just look at general patterns. These were the results for the tilapia at pH 5.3. And so for body position, what we looked at is basically normal, erratic swimming, lethargic, loss of equilibrium, or no body movement. For the breathing response, it was normal, gulping at the surface, slow gill movement or no gill movement, and then position in the water column was either the bottom or the middle or the top. And so the column, this first column, this is the initial treatment, then the recovery, and then this is the repeated treatment. And this is the time is along the x-axis. And so for tilapia, we can see that they were impacted almost immediately by 5.3, with almost all of them with no body movement. During the repeated treatment, it's pretty much the same, no body movement, so they were very heavily impacted. The breathing response was dominated by either slow gill movement or no gill movement, and that was pretty much, again, the same pattern for both treatments. And their position in the water column was essentially at the bottom. I didn't point it out, but this first column is kind of the pre-treatment behavior, and it's red, so those are normal, and at the bottom of them. And at 5.0, essentially the same results, except for this was almost, breathing response was almost all no movement, so it clearly impacted them. For guppies at 5.0, it's a little bit different, but still they were impacted pretty heavily. So initially, most of them showed no movement, but after about 48 to 50 minutes, a number of individuals started to recover. And so they resumed normal body position, and that pattern was kind of repeated during the second treatment. Most of them no movement, but about, 33% or so, 40% were normal. Breathing response, initially slow gill movement, or during the second treatment, slow gill movement or no gill movement. But again, there was a subset of them, about a quarter of them or more, a third of them that were normal. So they seem to be acclimating to the treatment. And then the position of the water column, somewhere at the top, somewhere at the bottom. And that was kind of mixed for the two treatments. Western mosquito fish wasn't too different from the mosquitoes. Again, they were impacted immediately, pretty heavily. There was no movement for most of them, a little bit of recovery, and then during the second treatment, about 40% of them were either normal or they were lethargic, and the rest there was no movement. So again, some acclimation. Breathing response, slow to no gill movement during the first, but again, half of the individuals were normal during that second treatment. And then position the water column. They were either at the top or the bottom. It was a little bit mixed, no real pattern. So the results from this quorum study, so this is the mortality. And tilapia were very heavily impacted. This was a pretty small study, so at 5.3, all 4 adults died. These adults, 6 of 6 died, and 8 of 8 juveniles died. So complete mortality for tilapia. This gave us hope for sure for the field treatments, that if we could attain these pH levels, and then we may be in pretty good shape. For guppies, it was mixed. There was survival. So 5 of 12 adults survived, 4 of 10 juveniles survived, and there was survival for western mosquito fish too, except for the juveniles. All of those died. For the opae'ula, 6 of 20 died, 14 of them survived, but the feeble shrimp, all 8 died. So this was not very satisfying in terms of results, because it didn't really inform us about what might happen to the more rare species, which is kind of what we were after. And so we don't know, but I guess silver lining on that is if there are tilapia in the pools and we're treating the pools, the chance of there being a shrimp in there is extremely rare, because they tend not to be present in pools with tilapia. And then at the arid snails, there was all 8 of them survived. So we're ready to try this in the field. So we're going to do pool in Hōnaunau, which is a park south of here. I always want to say that these are outstanding, beautiful parks, because they truly are. But it's another small park that protects major cultural features. This is the royal grounds, and this is the wall that separates the uhonua, or the place of refuge, which provided protection for folks during kind of warring times. And these yellow arrows point to 2 small fish ponds, and this is our study site, and I want to look at that a little bit closer. And again, these are the 2 royal fish ponds. They're on the royal grounds. This is the Great Wall, which is about 4 meters across, and it's kind of this expertly dry rock masonry. It's pretty stable, but it also is a little bit porous. And then this is the Makaloa pool that we treated, and the Makaloa pool is divided by two sub-pools called large and small. And at high tide, these are connected. At low tide, they're separate. But one thing I want to point out is that during very high tide events, the royal fish pond here and the Makaloa pond, they both come up against this rock wall. And both of these contain tilapia and western mosquito fish. Point of introduction is not clear, but for tilapia anyway, the 1960s or potentially even before. This shows the large subpool, giving an idea of what it was like before treatment. This is during the outgoing tide. These are all tilapia. This is a western mosquito fish. It's not a very good picture. it gives the impression, if you can see it, that there's not very many, but there are literally thousands in this pool. Something to point out are these breeding arenas, and those were the males build those for displaying for the females, and they seem to be required for reproduction. In their absence, after treatment, gave us some confidence anyway that the breeding was not taking place. If they would have been present, we would have been concerned. And you can just kind of see this sediment that people have been talking about, and it's up to 30 centimeters thick in some areas. Really quite amazing. So our methods were very similar to the aquarium trials, but on a much larger scale. We used the CO2 gas in kind of these 20-pound cylinders. We used these air stones that were probably about 1 meter in length, and they call them microdiffusers, so they're extremely fine bubbles. So 2 to 3 pairs throughout the pool. And our strategy was to place them at one end of the pool and then slowly move them across the pool, because we certainly figured that there was going to be some kind of a behavioral response and avoidance. At least we could concentrate the tilapia on one end of the pool. We did this during the receding tide, and our reasoning for that is, one, there would be less water in the pool, so you wouldn't have to treat quite as much. And also, we knew that the tilapia were going to go into the sediments when disturbed, so the idea was that it would pull the affected water into the sediments and better contact the tilapia. For about 2 hours for each treatment. I didn't say that, sorry. Bottom line is it was more difficult than anticipated to lower the pH, and the lowest that we reached was 5.7, but that did affect tilapia. And so during the first treatment, we ended up treating 4 times. The first one, we removed 439 tilapia, 1,024 mosquito fish. We quickly realized that there's so many mosquito fish in there, and they were not as affected as severely that for the resources we had, we probably weren't going to be able to remove mosquito fish, so we decided just to focus on tilapia. And the figure on the right kind of shows the body length. And we didn't measure the initial 434, but they were all about 10 to 12 centimeters in size. There were also some tilapia eggs. I mean, there was reproduction that had been taking place at the time pre-treatment. They're mouth birders, the females, and they would spit out the eggs when they were disturbed. And so we saw these little clusters of eggs, and we did the best we could to get them out. And the second treatment, about 5 weeks later, we removed 77 tilapia, and there were a bunch of them that were kind of on the small end of this the spectrum. And then on the 21st of November, about 3 months later, we removed 20 tilapia, and then another, about 2 months later, removed 3 more tilapia. And at that time, we were only getting large ones, and we were hopeful that those were the last 3 that were in that pool. Carter Atkinson on our team developed an eDNA assay for detecting tilapia as well as western mosquito fish and guppies in these pools, and so we kind of use that as an augmentation. It's extremely difficult to confirm eradication. Anybody that's involved in eradication studies in terrestrial environments, they bring in sniffer dogs and things like that. It's just the last thing you want to do is declare victory and walk away and then realize that you were wrong. So Carter developed this eDNA technique, and he and Malia did some testing to see how long DNA would persist in the water or in the sediments, and it seemed like water, it would degrade really quickly, a matter of just a few days. Sediments, a little bit longer, but by a week or 2, it seemed that there would be no detectable DNA in the water, and so that was important, and that informed the frequency at which you would want to sample. I'll let Carter talk about the methods. He's much more knowledgeable than I am as the geneticist, but the bottom line is that the eDNA data corroborated our observational data for up to 6 months that there were no tilapia in this pool. So at the end of that, we felt pretty confident that we had eradicated tilapia from that pool. So conclusions, it was difficult to lower the pH, again, more difficult than we anticipated. Why that is, it's not completely clear, but there's always a freshwater input, and also because the pool is actually very shallow, and so the residence time of CO2 in the pool was really very short. And so in this case, a deeper pool probably would have been easier. But it was still effective against tilapia, stunned them, challenging to collect. I can tell you all about that, or Malia can tell you all about that. Behaviorally, they're super challenging. They dive into sediments, they go under rocks. At the early stage, the juveniles mixed with mosquito fish, so they were really difficult to differentiate. Our strategy was, because we knew we didn't get all the juveniles because of those eggs that we decided, so we wanted to have a sampling or a treatment interval that was long enough so the tilapia could grow up to a certain size to differentiate them from the mosquito fish. But we really didn't want reproduction to take place because that would have defeated the purpose. So anyway, I think that worked out well. Western mosquito fish were less affected, but it did affect their behavior. They totally, but they avoided the CO2 and they tend to aggregate in little reservoirs or little channels away from it. So I think it could be used as a tool to help kind of drive them into nets and things like that. I think eDNA is certainly proved to be a useful screening tool to give us more confidence that if we don't see them, that they're truly not there. And then the knowledge of fish behavior is really important. And again, with tilapia, their behavior is very different than the Western mosquito fish. They do dive under rocks. They're really difficult. We're basically down hand feeling the bottom to try to get the last fish. If we wouldn't have done that, I think they would have survived because our pH was 5.7. They would have probably recovered and been just fine. And then, as I mentioned, the timing of reproduction is really important. You want to prevent. You want to do repeated sampling before they're able to reproduce. And I think with like guppies and Western mosquito fish, that's a real problem, with a little bit less so with tilapia. Finally, unintended consequences. There was just an algal bloom after removing of the fish. We hypothesized that there was some kind of a grazing release. It was always in the pool, just went unnoticed, but it's not necessarily a bad thing. It's a native vulva, but it was a phenomenon. Finally, I just want to mention that almost all research, you stand on the shoulders of people that are before you, and I want to acknowledge that we were not the first people to work on CO2 in anything pools in Hawaii. In mentioning Ann Brazier, Megan Leahy, and Barbara Siddell working at uhonua and Hōnaunau in the past, Dina Sidar at Keikaha State Park, Sally Bubers and Kailua-Kona November at Koloko, and I apologize if there's others who have done CO2 work and I'm not aware of it, but anyway, like I say, we stand on the shoulders of others. So thank you very much.